Welcome to Roadmap to Residency Evidence-Based Applications. Hello everyone, I am Abhiras, and in this video we will be discussing in detail about regulation of gene expression. So talking about bacterial metabolism, so bacteria need to respond quickly to changes in their environment. So if they have enough of a product and they need to stop the production. Why? Because it is a waste of energy to produce more. And how? To stop the production of enzymes or for synthesis. So if they find new food or energy source, they need to utilize it quickly. Why? Because the metabolism, growth, and reproduction part. And how? Because they start production of enzymes for digestion. So remember regulating metabolism. So feedback inhibition, which is the product acts as an allosteric inhibitor of the first enzyme in the tryptophan pathway. Okay, you can see that tryptophan, it if there is a lot of tryptophan, then it just inhibits the first enzyme in this pathway. But this is a wasteful production of enzyme. So gene regulation, so instead of blocking the enzyme function, we block the transcription of genes for all enzymes in the tryptophan pathway. It saves energy by not wasting it on unnecessary protein synthesis. So this is what a bacteria does. So gene regulation in bacteria, so cells vary amount of specific enzymes by regulating the gene expression. They turn the genes on or they turn the genes off. The turn gene off example, so if bacterium has enough tryptophan, then it does not need to make enzymes used to build tryptophan. If bacterium encounters new sugar like lactose, then it needs to start making enzymes used to digest lactose. So that is the gene turn on and gene turn off mechanism. So bacteria group genes together. So number one is a concept is about the operon. So the genes grouped together with related functions is called as an operon. Promoter is the RNA polymerase binding site. An operator is a DNA binding site of a repressor protein. So how can these genes be turned off? So by repressor protein, which can bind to the DNA at the operator site, which can block the RNA polymerase, or which can block the transcription. So operant model. So operator, promoter, and genes, they control serve as a model for gene regulation, right? And the repressor protein, they turn off the gene by blocking the RNA polymerase binding site. So when excess of tryptophan is present, it binds the tryptophan receptor repressor protein and triggers the repressor to bind to DNA. Then it blocks or represses the transcription. So what happens when tryptophan is present? We don't need to make tryptophan building enzymes, right? We need to stop. So then there is the inducible operon, or in case of lactose, for example, when lactose is present, it binds to the lac repressor protein and triggers the repressor to release the DNA. And this DNA then induces the transcription to produce the enzyme which will help the bacteria to digest the lactose. So what happens when lactose is present? We need to make lactose digesting enzymes, right? So operon summary is that it usually functions in anabolic pathways for synthesizing in products. When in product is present in excess, cell allocates resources to other users. Inducible operand is when it usually functions in catabolic pathway, digesting nutrients into simpler compounds, and it produces enzymes only when nutrient is available. Now let's talk about the positive gene control. So it occurs when an activated molecule interacts directly with the genome to switch transcription on. Even if the lac operon is turned on by the presence of allolactose, the degree of transcription depends on the concentration of other substrates. The cellular metabolism is biased toward the utilization of glucose. Some operons are also subject to positive control through a stimulated protein, such as catabolite activated protein and activator transcription. When glucose is scarce, the catabolite activated protein is activated by binding with cyclic AMP. Now, activated CAP or catabolite activated protein attaches to the promoter of the lac operon and increases the affinity of RNA polymerase, thus accelerating the transcription. Positive gene regulation. So if glucose levels are low along with overall energy levels, then cyclic AMP or CAMP binds to the CAMP receptor protein, then which activates the transcription. But if glucose levels are sufficient and CAMP levels are low, then the CRP protein has an inactive shape and cannot bind upstream of the lac promoter. So the big questions are, how are genes turned on and off in eukaryotes? How do cells with some genes differentiate to perform completely different and specialized functions? This is through gene regulation. So there are different points of control. So the control of gene expression can occur at any step in the pathway from gene to functional protein. It can occur while packing or unpacking of DNA, in the process of transcription, in the process of mRNA processing or transport, in the process of translation of protein, in case of protein processing, or in case of protein degradation. The points of control can be anywhere. So DNA packing as gene control. So degree of packing DNA regulates the transcription. If the DNA is tightly wrapped around histones, there is no transcription and the genes are turned off. DNA methylation. So methylation of DNA blocks the transcription process. So there is no transcription. The genes are turned off. Histone acetylation. So acetylation of histone unwinds the DNA, which means that DNA is now loosely wrapped. So if the DNA is tightly wrapped, then it stops the transcription. But if the DNA is loosely wrapped, then it enables the transcription, which means that now the gene is turned on. So let's talk about epigenetic inheritance. So although the chromatin modifications just discussed do not alter the DNA sequence, they may be passed to future generation of cells. The inheritance of traits transmitted by the mechanisms not directly involving the nucleotide sequence is called as epigenetic inheritance. So transcription initiation. So the control regions on the DNA, like promoter and enhancer. So the promoter is a nearby control sequence on the DNA. The binding of RNA polymerase and transcription factors occurs here, and it is a base rate of transcription. Enhancer is the distant control sequencer on the DNA, where the binding of activated proteins take place, and yet it is the enhanced rate of transcription. So model for enhancer protein, it is there like enhancer DNA sequences which are in the distant part and the promoter sequences which are in the near part. Again, now this is the basic picture of transcription complex, not so much important. We can see the activated proteins, the regulatory proteins bind to the DNA at distant enhancer site, and they increase the rate of transcription. That is, it enhances the rate of transmission, and then it has the enhancer sites, which are the regulatory sites on DNA distant from gene. The post-transcriptional control is when there is alternative RNA splicing. The variable processing of exons creates a family of proteins. Lifespan of mRNA determines amount of protein synthesis, and mRNA can last from hours to weeks. So the blocking of initiation of translation stage, the regulatory proteins attached to the 5' end of mRNA, it prevents the attachment of ribosomal subunits and initiated tRNA. It blocks the translation of mRNA to protein. Protein processing and degradation. So the folding, cleaving, adding sugar groups, targeting for transport, they are all are present in case of protein processing. But the ubiquitin tagging or the proteasome degradation, they all lead to the protein degradation. So ubiquitin is also called as the death tag. They mark the unwanted proteins with a label, and then these are engulfed by proteasomes. So proteasomes are again the protein degrading machine, the cell's waste disposer, and they break down any proteins into 7 to 9 amino acid fragments. So RNA interference. So small interfering RNAs are the short segments of RNA with 21 to 28 bases, and they bind to mRNA and they create a sequence of double-stranded mRNA, and they are the death tag of mRNA, and they cause gene silencing. So gene regulation, again, it can be through transcription in case of DNA packing and transcription factors. It can be in post-transcription process like mRNA processing, splicing. It can be translation process like the blocking the start of translation, or it can be in the post-translation process in protein processing and protein degradation. So the molecular biology of cancer, first of all, let's talk about oncogenes. So oncogenes are the cancer-causing genes, and proto-oncogene is a normal cellular gene. So how the movement of DNA, the chromosome fragments, they have resolved incorrectly, and it increases the number of copies of proto-oncogenes, and that's how the tumor genes, they just go on dividing. And also the tumor suppressor genes are the changes in genes that prevent the uncontrolled cell growth. And if there is defect in the tumor suppressor gene, then there is progression of cancers. So cancer cells result from a series of genetic changes in a cell lineage. The incidence of cancer increases with age because multiple somatic mutations are required to produce a cancer cell. As in many cancers, the development of colon cancer is gradual. Thank you for watching. Keep studying hard.